Okay, hello. This is my video about the cell cycle. So it's Anna, Paige, and Brooke. So we're just gonna get right into it, ladies. So right here we have a quick overview of the stages of the cell cycle. So the cell cycle has a complex set of stages before going into the actual cell division. The two main steps are interphase and mitotic phase. So interphase goes first over here, and then the mitotic phase goes second. And then so interphase prepares a cell to split through three phases, including G1, S, and G2. G1 is when the cell grows, and towards the end, there's a checkpoint. A checkpoint is a series of tests that the cell must pass before proceeding into the next phase of the cell cycle. Once it passes the checkpoint, it moves on to the S phase, where the DNA is replicated. After this, the cell grows more in the G2 and goes through a separate checkpoint right before entering the mitotic phase. The mitotic phase consists of two stages. So the first one is mitosis, and the second one is cytokinesis. Mitosis is the splitting of the nuclear membrane and DNA into two sister nuclei. During mitosis, there is another checkpoint. This checkpoint makes sure all chromosomes are attached to the spindle before anaphase begins. Then cytokinesis is the splitting of the cytoplasm and the rest of the cell. Each checkpoint is run by proteins called cyclins and cyclin-dependent kinases. Cancer occurs when the cell cycle is interrupted and the cells continue to divide despite external factors that would cause other cells to stop dividing, which is why we have cancer up here as its own separate thing. So first, we're going to go into these two pictures that kind of show what the cell cycle is. So in the picture on the left, this shows each stage of the cell cycle and the amount of time each phase takes, or each stage. As shown, interphase takes the longest because the three steps are preparing the cell to actually split. Then, the picture over here on the right shows how the cell is coming apart, with the two separate nuclei forming with two completely sets of DNA in each new daughter cell. And around here, the green, you can see the membrane. So next, we're going to go into the actual checkpoints and what they actually do. So the cell cycle is regulated by a series of checkpoints, which is G1, um, S, and G2. So the checkpoints are made up of proteins called cyclins and enzymes, which are the cyclin dependent kinesis. Um, so these kineses activate and inactivate certain proteins by phosphorylation. If the cell does not meet a certain checkpoint or pass the test, the checks wait. So if the cell does not meet a certain checkpoint or pass the test the checkpoints offer, the cell will go into G0 phase, which is over here, as it says here, resting state, and it pretty much means that the cell can't keep going and will not split. An example of cells that do go into G0 or remain in this phase are nerve or muscle cells in the human body because they obviously never divide. So, the first checkpoint right here, which is G1, checks for nutrients, growth factors, and DNA damage. The next one is S, then it goes on to G2, which is the other checkpoint that checks for cell size and DNA replication, since in the S they're replicating. And then finally, right before the cell splits, you have the M checkpoint, and it checks for to make sure all the chromosomes are attached to the spindle. Next, we have the PDGF and the MPF, which are internal and external factors that um, affect the cell division. So first we have PDGF, which stands for platelet-derived growth factor, and it is required for cells in human bodies to divide because they need it to pass the G1 checkpoint. So the PDGF is a stop-or-go signal, which is needed for the cell to go onto the S phase, which comes right after the G1 checkpoint, or into the G0 phase, which is obviously when it stops and it doesn't keep going and it doesn't split. Then another signal that we have over here is the MPF, or the mitosis promoting factor, and it is needed to pass the G2 phase. This allows the cell to enter the mitotic phase. These are two types of internal signals that are sent in order for the cell to split. So both of these are internal. Then we have how all of this, the entire cell cycle, and an interruption of it actually causes cancer. So, cancer cells are produced when there is an interruption in the cell cycle. Most cells need chemicals called growth factors in order to go through the cell cycle. When cells do not need to divide anymore, the growth factors are depleted and they stop replicating. Depleting obviously meaning that they are taken away or they're not there anymore. So cancer cells continue replicating even when the growth factors are gone. They also ignore the normal signals that control the cell cycle, so they grow uncontrollably. When there is one change in one gene or more, which is an example, this would be like a mutation that changes the function of their proteins, which causes a faulty cell cycle control. And when there's a faulty cell cycle control, it means that the cells continue and stop growing whenever they want. So there really is nothing that stops them or keeps them growing. They just pretty much do whatever they want, and in most cases, they obviously keep growing. Um, so, cancer cells also grow out of control because they have no recognition of cellular crowding, also known as density dependent inhibition. Cancer cells can also become metastasized, which means cells start to move from one part of the body to another and spreading to new areas. This is caused by cancer cells not needing anchorage dependence, or when cells need to be attached to a solid surface in order to thrive. So, pretty much, for example, right here, you can see that they keep growing on top of each other instead of just one layer. So that kind of shows the whole what do you call it? The density dependent inhibition. And then obviously, as stated before, they detach from themselves because they don't have the anchorage dependence, so they kind of just spread wherever they want. And yeah, that's all. Thank you.